Thank you so much for the great introduction. I'm definitely honored to be here to tell you guys how we could use artificial intelligence for drug discovery. How many of you guys in this room heard of Erome's law? Okay, I can see a few hands. That's basically Moore's law spelled backward. In Moore's law, we expect that the number of transistors in a computer doubled every 2 years, so the capacity of computers increases. Erome's law is the opposite, and what we have been observing in pharmaceutical industry, that the efficiency of the drug discovery and development decline by time. Right now, it takes over $25 billion to discover a compound, and the rate of failure is over 90%. So we are in a desperate need for innovative technologies that could improve. Even a slight bit of this efficiency can have a significant impact. But why AI, and why now? AI has been around since 1950s. In fact, it survived 2 winters. The winters that happened for AI was mostly because the computers were not efficient enough. The capacity of the computers were so limited that they couldn't stand up the hype. So there were so many critics that didn't believe that we could develop a machine that could be more intelligent than human. But that changed as the technology advanced and the capacity of the computer increased. We had some landmark breakthroughs that happened in early 2000. In 2006, University of Toronto developed deep learning. In 2011 and 12, the application of deep learning and running them in GPUs have significantly improved those algorithmic predictions. And we only recently saw that IBM and also Go have been competing with champions, leader champions of different games and overcoming them. So there is more and more interest in the application of AI. And we have been seeing the application of AI in many industries, considering in automation, finance, self-driving cars, and robotics. But is pharma ready for AI? Pharma has been relatively one of the late adopters of using AI technologies. But because of these trends and growth and interest in this field, as well as the need for innovative technologies, we have been seeing the application of AI in drug discovery. In fact, if you look at the papers that have been published in PubMed for using AI for drug discovery, since 2010, we have seen any more papers than any other prior race combined together. We have also observed the amount of funding that has been going to AI companies, as well as the number of collaborations that have been happening now between pharmaceutical industries and AI companies have significantly grown. There are different players that are also forming this market. We've seen many pharmaceutical companies, large pharma are playing this role. There are lots of AI startup companies that are growing. We have seen many investors, especially the tech giant investors, that now they're shifting to the AI space and they're trying to invest more in AI-driven drug discovery companies. And even tech corporations that are moving to this space, including Apple or Google. As I mentioned, AI has different applications, but where AI can have significantly play a role, AI is a tool. So we can use it smart enough to make sure it could help us to improve the efficiency of drug discovery and development. There are different parts of drug discovery that we could use AI, from phenotypic screening, lead identification, lead optimization, and in clinical trials as well. But perhaps what AI can help us the most is to understand human biology. If we really understand at an individual level what happened in our body, in molecular level, that lead to the disease, we could develop effective therapeutics. Many of the major failures that have been happening so far, because we really don't know what's happening. We are doing guesswork. Particularly for Alzheimer's, you've been hearing all these recent failures. Most of them has caused, if we really know what happened in the neurons that start the degenerative process, it will really help us to discover the effective therapeutics with a higher success rate. But that's not so easy either, because there are so many elements that lead to developing a disease. It's not just our genes. We have started sequencing human genome. In 2003, we thought we were going to solve and cure all the diseases by sequencing human genome. But now we know that the reality is far from that, because only less than 10% of diseases have a strong genetic cause. So genes are important, but not the only thing. Our diet and lifestyle have a significant role in developing diseases. We are hearing more and more about the role of microbiome in our health and our developing diseases. In addition to that, even our social network has such an impact. The people who we interact with have impact in our life. There was actually a paper came out a few days ago that was showing how our microbiota transmitted between our social network and our families. So it's not that simple. There are so many factors that helping us that lead to developing a disease for each patient. And that leads to this problem that the biological data is, the matrix of biological data is sparse. For each patient, we have some set of data, but really to completely understand what happened, for each one of these patients, we need more and more data. And that's one of the limitations that currently exists and one of the challenges of using AI effectively for drug discovery. This data is also, there is this hype that the data is overestimated. We think there is lots of data, there is big data, but that's not the case. I want to bring this example. AlexNet was one of the algorithms that developed in 2012 that significantly improved the state-of-the-art image analysis and image recognition using GPU. AlexNet had to use 15 million label data in 22,000 categories to reach that performance. But we don't have that much data in biology. We don't have all these labels. If we have all the labels, we've already cured so many diseases. So how we could use AI more efficiently to overcome this problem and overcome these issues of leveraging AI for drug discovery? One of the things that we could do to bring more set of data to the picture, collect more data from each person, and being able to integrate all these data sets together to understand the big picture of biological processes. And one of the things is me and my team at Revivement, we are excited to bring more data about metabolites. Metabolites are any molecules that are super small in their size, like sugar, glucose, fats, cholesterol, things that we hear all the time in our health and diet, right? It's been known that there is, in fact, there are over 100,000 of these metabolites in our body. They are downstream of DNA, so they carry information from our genes, and they also can reflect what our lifestyle has an impact in our health, because they are involved in our metabolism. So our goal is to bring more data about these metabolites to the picture, combine them with all the other data that we have about our genes and RNA and proteins to understand big biological picture. But this problem was not that easy either, because there are over hundreds of thousands of these metabolites in our body, and each one of them is different from the other one. So it's actually very hard to clearly characterize all those 100,000 for each patient to understand what happened in that disease. And people usually call this as the dark matter of metabolomics or emerging crisis, because from tissues or biofluid of patients, we could measure some of the metabolites using mass spectrometry. We measure a mass of a molecule, but we don't know what those things are. We know there should be in our body, but we can't clearly characterize them. And current technologies are only able to characterize hundreds of them. As I mentioned, there is over 100,000 these molecules in our body. And I usually tell people, when we only know the data about 100 molecules, we don't need to use AI. You could use just Excel and analyze these data sets and get the same insight. And it's estimated, if you want to solve this problem experimentally, it could take decades and billions of dollars. So that's one of the areas that we could see experimental solution may not be a solution. And that's why we developed an AI platform to overcome this problem. We leverage the exponential growth of biological data and then to build a really comprehensive database about metabolites, and then leverage that database using our AI algorithm to figure out the identity to clearly characterize metabolites, and then use that for drug discovery purposes. So what we are doing usually, we start from tissues or blood of patients, and we do mass spectrometry. We measure masses of molecules. We measure tens of thousands of them. But as I mentioned earlier, we don't know exactly what they are, because there could be 10 metabolites with the exact same mass. I have this dummy example. If it's a mass of 180, it could be either glucose, galactose, or fructose. they all have the same mass but different functions in our body. What we are doing instead, our AI algorithm consider all those ambiguities and mine that network, that mega network I showed you guys earlier, to find a way that how those metabolite masses that are dysregulated in a disease are connected to each other that lead to the disease. And through this connection, we are able to infer, okay, this 180 here is glucose because glucose is connected to the disease network. So we are able now to identify those metabolite masses that was not possible before. And more importantly, finding these novel mechanisms, so how those molecules are connected to each other that lead to the disease. Because this novel understanding about the biology then help us to discover therapeutic targets or existing drugs that are linked to those pathways and processes. And as I mentioned earlier, our goal is to bring different type of data together because each type of biological data can give us a single view of biological processes. And in order to whole understanding the big picture, we need to combine different type of data together. And actually what we show was that if we look at metabolomic data, we could get some insight. And we also look at some other type of omic data, like proteomics, and we get different insight. And we actually did a systematic comparison. We saw the pathway that we infer from metabolomics was really not overlapping with the pathways that we infer from proteomics data. But what we observed was that when we're bringing these two type of data sets together, we capture the majority of the insight from each type of data set separately, but we also find this new thing. So I show you with the red circle here. We find new pathways that are only able to infer them when we bring in different type of biological data sets together. So I don't go to the details, but this was a work I did during my PhD at MIT, and we experimentally validated all these findings and published it in Nature Methods. I'll be happy to answer questions, but for the sake of time, I will just stay with the brief overview of these processes. And our goal is that at ReviveMed, by collecting more and more data about metabolites and understanding how changes in metabolites lead to other diseases and connection of this type of data to other type of biological data, our algorithms will get smarter and smarter to discover the right therapeutics for the right patients. And we are excited to be in this great area of the intersection of AI and metabolomics, and our goal is just to create impact for patients and saving people's lives with every line of code. And thank you.